Hi, Alan. So this is Alen Suljic from Ljubljana in Slovenia. He's doing his PhD at the moment, I think. Is that right, Alan? Yep. And he's going to talk to us about spike screen, a method for monitoring SARS-CoV-2 variants. So over to you, Alan.
Can everyone see the slides?
Yes, we can.
Yes. So thank you, Dr. Bailey, for a lovely introduction. And I would also like to thank the rest of the organizing team for inviting me to this virtual seminar series of the UK-ICN. So my talk focuses on challenges and potential solutions of variant surveillance in limited capacity setting during the COVID-19 pandemic, from the appearance of early lineages to emergence and disappearance of the variants of concern. So, after the first reported case of SARS-CoV-2 in Slovenia on March the 4th, 2020, the number of cases began to rise and at certain points overwhelmed the diagnostic capabilities of laboratories. Using only research sequencing capabilities, we were not able to characterize a sufficient number of samples for timely forecasting of the epidemic. We tried to partially address the problem by using additional sequencing capabilities provided by ACDC, but still couldn't get sufficient numbers of samples. Although we managed to increase diagnostic capacity by the end of 2020 with higher throughput sequencer, the emergence of SARS-CoV-2 variants revealed another deficit that underscored the need for rapid and efficient genome sequencing. So this image shows in gray bars the total positive samples in each given week, and red bars indicate the number of sequenced samples with our routine complete genome sequencing program. So mutations are a natural part of the replication cycle of any RNA virus, resulting in evolution of variants with increased transmissibility, disease severity, and vaccine escape due to immunological memory. For this reason, the real-time genomic surveillance is important for understanding potential public health impact and evolution of the virus. The surveillance using next-generation sequencing provides us with a reliable and accurate information on the prevalence of the circulating variants. In order to control the spread of the virus, to develop vaccines, to inform public health policies, and to direct epidemiological interventions. However, with every surveillance effort, the result largely depends on the sampling strategy and sample size, of course. So, in this figure shows the number of samples that we were able to characterize using the complete genome sequencing, shown in gray bars. Based on the weekly numbers of positive samples, we calculated the sample sizes needed in order to reliably detect variants circulating in population at different levels. So at 1% proportion, shown in green line, 2.5% proportion, shown in blue line, and 5% proportion in shown in red. We can observe that only with complete genome sequencing, we were able to perhaps ensure the detection of a circulating variant at 5% proportion, seen here in the first two waves, characterized by dominated by B1 25817 variant and the subsequent alpha wave, and then when we employed the higher throughput sequencer, we were able to reach 2.5% thresholds using the complete genome sequence. So the overall sampling strategy for the routine complete genome sequencing consisted of removal of low viral loads, duplicate and follow-up samples. So samples were considered duplicates if they belong to the same individual who already tested positive in each week. The number of sequence samples were then selected based on the sequencing capacity of the available sequencer. And in some cases, in many cases actually, the number of positive samples exceeded the sequencing capacities, and that's why we opted for random sampling in order to address the selection bias. So in order to meet the lower detection limits described earlier, we had to look for new, innovative cost, logistics, and labor-effective strategies for screening more samples. So in order to increase the number of surveyed samples, we developed a strategy called spike screen. Alongside the samples already sequenced by CGS, we subsampled an additional portion among weakly positive samples that were not already a part of the complete genome sequence. The samples, this subsample, were pooled according to the available metadata, so geographic information and the demographic information. So the samples were pool based. So in each sample, each pool covered 1 or 2 days of the epidemic. There were approximately 400 samples per pool, and to ensure comparable read depth per sample, the pools were arranged according to their respective CT values. So short sequencing libraries were then prepared according to the Arctic protocol, with one exception, that we selected and amplified just the spike gene portion of the SARS-CoV-2 genome, and we managed that by using a distinct portion of the Arctic primers. The full sequencing data was then analyzed for the mutation frequencies using the variant analysis. The bioinformatic pipeline consisted of the analysis priorly. So at the level of sequencer, there is some analysis being done. The subsequent reads were then trimmed by quality and length. The reads were then mapped to the reference genome. The variants were called using IVAR, and the subsequent formal analysis was conducted on this data. So for read mapping, we used Bova-Amin with some help from SAM tools. The variant calling was performed using IVAR. We opted for frequency threshold at 1% and with minimum read depth of 10 bases. The lineages were then designated using the fresh version of Pangolin. The formal analysis was done by R, and the mutation frequencies, the characteristic mutation frequencies were obtained using outbreak info. So in this image shows the additional capacity boost provided by the spike screen. Using spike screen, we sequence additional 825 pools of amplified spike gene, encompassing a total of 29,000 samples. This approach allowed us to expand the surveillance of the circulating variants by an additional 23,000 samples, or 55%, that were not included in the routine complete genome sequencing, while that's requiring only 3% of the resources and cost in manual play. We can see that here in the first two waves, we were able to, so in the beginning of 2021, we were able to reach the goal of 2.5% proportion threshold using the spike screen. And in later 2021, we were also able to reach the 1% proportion threshold. You will notice that I omitted the 6,000 samples, which were sequenced with both approaches, and we will find a use for them a bit later on. So we've seen that we can get the sufficient numbers of sample sequence using the spike screen, but does it work? How does it work regarding the mutation frequencies? So to demonstrate the efficacy and reliability of our pool sequencing strategy, we tracked the characteristic mutations in the spike gene between the transitions of the major variants. So this figure shows the variant dynamics of B1-25817 variant. That's in panel A. Panel B shows the alpha characteristic mutations. C shows delta, and D omicron. The black bolded line is from the complete genome sequencing data and represents the ground truth, let's say. And colored lines represent the surveilled mutations obtained by spike screen. So in the panel A, we can see that the color lines almost perfectly follow the populational variant prevalence, with the exception, of course, of D614G mutation, which was present in the earlier lineages and is basically consistent throughout the entire time course. The same dynamics, or the same mutual dynamics, could be observed between the alpha, delta, and omicron lineages. If you really pay attention, in the panel B, you can spot that mutational frequencies obtained by spike screen showed an earlier signal prior to that obtained from the complete genome sequence. So yeah, further investigation of the reliability of the spike screen strategy led us to examine the relationships between the pools with known lineage composition and the monitor frequencies of characteristic mutations in the spike gene in these pools. So the left panel shows a pool of samples with known lineages, and they were established using complete genome sequence. On the right, we can see the frequencies of mutations obtained by spike screen. So you will see that some mutations are not specific. That would indicate that that mutation is harbored by multiple lineages and therefore cannot serve as a marker mutation. So two tests of spike screen, the first two tests, was to detect the emerging threats of alpha. The use of specific PCR test for H69B70 deletion was not quite adequate, since the predominant variant at that time in Slovenia was, as said earlier, B1-25817, which already harbored that deletion. So for this reason, we relied on spike screen to monitor frequencies of other characteristic mutations to detect and monitor the spread of alpha. So in this case, we can see a detection of the characteristics mutations both for alpha and beta variant of concern using the spike screen. Along that, we can also see that two of the earlier lineages, so B1 160 and B1 221, we can observe that we detected also the characteristic mutations of these early lineages. The same thing can be demonstrated at the example of early lineages, where the predominant B1 25817 variant is seen, and a detection of low prevalence eta variant of concern. We can also observe that the frequencies of V772I mutation and N439K follow the frequencies are quite linear to the exact percentage of the lineage in the pool. Yes, that will be. The same thing could be demonstrated in Omicron, when we can see the same linearity between the frequency of the characteristic mutations and the true composition of the pool. So to really demonstrate that the spike screen strategy can reliably assess the percentage of given variant based on the characteristic mutation frequency, we performed a linear regression modeling for the P681H mutation that we saw that was very stable in the alpha variant. So we can observe nearly linear relationship with coefficient of 0.83. That is so close to 1 that we can say that the increase in the P681H mutation in the pool, that the rise for 1% of this mutation directly corresponds to the rise in 1% of the alpha variant in that pool. If we look at the adjusted R-squared metric, that shows 83%, which indicates that the mutation frequencies explain 83% of the variation in the percentage of the variant in the pool. So that indicates that very few additional factors influence this dynamics. We also found two outliers, here colored in red, but we traced them to the higher variance in CT values in the set pool. We actually didn't have the information, so all pools sequenced by spike screen, we didn't surveil everything with the complete genome sequence. So in some cases, like in these two examples, we can see that approximately 10% of the pool lineage, constellation was unknown. So only looking at the left point of the left panel of the first plot, we would not expect any other mutations than the one belonging to B1 25817, B1 258, and B1. However, on the right panel, we can clearly see the detection of the characteristic mutations belonging to alpha variant. So we can quite reliably say that this pool on the left contains at least one sample belonging to alpha variant. And the same example could be seen in the high alpha predominance setting in the pool, and with the detection of few delta samples in the pool. So our experience with spike screen, it enabled us to provide the public health policymakers with timely and reliable information on the course of the pandemic. it was said that the detection at the 2.5% resolution corresponds to about 7 to 8 weeks of forecasting before the variant becomes dominant. This is the assessment done on the growth rate of alpha with, I think, growth rate of 50% each week. We show that pooling allows scaling up of surveillance efforts, especially in situations where rapid characterization is required in large outbreak, or in countries or territories or settings with limited sequencing resources. The employment of spike screen actually allowed us to detect and contain a few high-profile variants, such as beta, eta, and gamma. Due to rapid detection and deployment of quarantines and complementary epidemiologic actions, the epidemiologists were able to quell the initial infectious momentum of these high-profile variants. So we speculate that a similar strategy could theoretically be applicable to population surveillance of any viral pathogen. So the selective amplification of target regions could be rapidly and reliably deployed in severe outbreaks or in settings with scarce sequencing resources. By choosing the appropriate target for different viruses, obviously, and clever use of bioinformatics tools, such approaches could aid in bridging the gaps in the global sequencing capacities. There's another advantage in sequencing the shorter section of viral genome, and that is the reduced computational power requirements, which is an important consideration in any bioinformatics workflow. This also represents a tremendous advantage for lower income countries where access to advanced computational infrastructure is, well, limited. So in summary, spike screen is a rapid, accurate, and cost-effective way to detect and track the emergence and abundance of SARS-CoV-2 variants. This approach is capable of monitoring a large number of samples in settings with limited sequencing capacity. It can serve as a background information for informing public health policy planning, treatment choice, or application of vaccination. Our results indicate that spike screening can successfully complement the individual case complete genome sequencing strategy by sacrificing less informative parts of the gene. So this stuff was only possible because of hard work and efforts from many people included in the diagnostic and research process, ranging from supervisors, laboratory experts, bioinformatics specialists, dedicated students, collaborators, and financiers. So yes, I would end here.
Lovely. Thanks for such an interesting talk, Alan.